We will continue our introductory material with a lecture on gastrointestinal physiology. After this lecture, you should be able to explain what makes an anaerobic environment and why anaerobes cannot live elsewhere. You should be able to explain also other common characteristics of the gastrointestinal habitat. You should be able to identify gastrointestinal adaptations and their relationship to diet and microbial fermentation, and you should be able to distinguish between different types of herbivory and the merits of each type. Your reading will be chapters 6 and 7 in the textbook. There are several kinds of anaerobic environments, like the gastrointestinal tract, silage, sediments, some deep sea regions, and even inside soil particles and other microenvironments. We'll discuss the gastrointestinal tract most in this class, and we'll also briefly talk about silage. These environments are unified by having no oxygen and also having a low oxidation reduction or redox potential. You'll recall from our last lecture that obligate anaerobes are those that cannot grow in the presence of oxygen. And in this figure, obligate anaerobes are represented by tube 2. That is, organisms that can grow at the bottom of the tube where there is minimal or no oxygen. As we'll discuss later, this figure is not really correct. Even in the absence of oxygen, strict anaerobes would not be expected to grow. They need more than just no oxygen. Oxygen is toxic to cells. There are two different forms of toxic oxygen. One is superoxide, which you can see up top, and another is hydrogen peroxide, and you can see how each of these forms from oxygen. Many cells have defenses against these toxic forms. For example, superoxide dismutase converts superoxide to hydrogen peroxide. Catalase converts hydrogen peroxide to water and oxygen. Many cells and organisms have these defenses, but not most anaerobes, and that is one reason why oxygen is toxic to anaerobes. Low oxygen, though, is not enough for anaerobes. Their environment is still too oxidizing, even without oxygen. And to understand that, we have to refer to the redox potential. You can recall the redox potential from your introductory chemistry classes. We have here on the right, redox potential, where higher values represent a more oxidized environment and lower values represent a more reduced environment. And we can see that water in equilibrium with the normal atmosphere of 0.21 bar oxygen has a pretty high redox potential. It's about 0.7. The rumen has a very low redox potential, around negative 0.3. We could never create an environment without any oxygen, but let's say that we could create one with almost no oxygen. We could create a nearly oxygen-free atmosphere, one that has only one molecule per liter. Not 1 mole per liter, but a single molecule per liter. And we can see the redox potential of a solution equilibrium with that atmosphere is still too high. It's about 0.3 to 0.4. What allows anaerobic environments to have such a low redox potential is the presence of reducing agents like hydrogen we see here. A solution equilibrium with hydrogen has a very low redox potential, slightly lower than that of the rumen. Low oxygen is not enough. These reducing agents have to also be present. Why is oxygen and a high redox toxic to anaerobes? It's not really known, but there are two hypotheses. Toxic forms of oxygen oxidize cellular compounds, like thiol-containing enzymes and other compounds, iron-sulfur proteins, and flavoproteins. These are all reduced cellular compounds that are deactivated when they're oxidized. High redox potential could deplete the cell of reducing power like NADPH that's used during biosynthesis. Rather than being coupled to biosynthesis, NADPH could simply be oxidized to NADP. These are two hypotheses, but there have not been many controlled experiments to test them. We'll shift our discussion back to the gastrointestinal tract, which is the site of diet digestion and microbial fermentation, and thus important for discussion of gastrointestinal microbiology. One chief characteristic of the gastrointestinal tract is that it's largely anaerobic, as we have discussed, but there are other important characteristics that we will discuss on the next slide. This table reports in detail the physical, chemical, and microbiological characteristics of the gastrointestinal tract, specifically fermentative compartments in the gastrointestinal tract. You don't need to know all these details. The purpose of this table is to give you a general appreciation of the characteristics. For physical characteristics, we see that pH is around 6. The redox potential is low, as we saw earlier, and the temperature is around physiological. We will not focus on the other characteristics shown. For chemical characteristics, we see the major VFA are acetate, propionate, and butyrate. Lactate can also be present at lower concentrations. Together, the VFA and lactate have a high concentration at roughly 100 millimolar. Amino acids and oligopeptides are present, but at low concentrations, less than 1 millimolar after feeding. Ammonia is present at somewhat higher concentrations, at 2 to 12 millimolar. Soluble carbohydrates are present in low concentrations, less than 1 millimolar. In contrast to these soluble nutrients, insoluble polysaccharides like cellulose, hemicellulose, pectin, and lignin are present at high concentrations. The table simply says they are always present. For microbiological characteristics, we see fermentative compartments have bacteria, ciliate protozoa, anaerobic fungi, and bacteriophages, with the concentration of bacteria being highest. We will discuss these characteristics in more detail in future lectures. In addition to discussing these general characteristics, we want to discuss how anatomy in the gastrointestinal tract is adapted to diet. And we'll go through this for carnivores, insectivores, frugivores, as well as omnivores and herbivores. We will review gastrointestinal tracts from several animals according to these diet types. You'll need to identify the stomach, the small intestine, which is also known as the midgut, the cecum, which is part of the hindgut, and the colon, which is also part of the hindgut. We can see here the stomach, the small intestine, the cecum, and the colon in the adult human. I will not point out these structures on each of the following slides. You'll have to be able to identify those yourself. There are other structures that you'll need to be able to identify, and these will be pointed out to you in each of the following slides. The carnivores, insectivores, and frugivores are united by having little microbial fermentation, and we can see examples of these in the figure at the right. We have a cat, which is a carnivore, a bat, which is insectivorous, and a squirrel monkey, which is both a frugivore and an insectivore. I was not able to find a gastrointestinal tract of a strict frugivore. For these, the stomach is glandular, and it's very simple. It looks pretty similar to our own stomach. All have a short hindgut, and this hindgut is often indistinct from the midgut. For example, in the bat, it's very hard to distinguish where the midgut ends and where the hindgut begins. For the cat, the hindgut is a bit enlarged, and for the squirrel monkey, there is a small cecum. There is no need for complex architecture in general because the diet is digested very quickly. For example, in some frugivorous bats, the diet is passed within 20 minutes of consumption. With omnivores, we start to see some microbial fermentation and some elaboration of the gastrointestinal tract. We have the black bear and the possum here as examples. They still have simple stomachs. They look pretty similar to our own. However, the midgut and hindgut are oftentimes longer. They're of variable length. The midgut is quite long in the black bear. You can see how long the midgut would be if we unraveled it. In the possum, the hindgut is long and it's enlarged. And the midgut and hindgut are often longer or better developed than in carnivores, insectivores, and frugivores. This longer and more complex tract reflects the more complex and difficult to digest diet. Plant material is difficult to digest, it must be fermented, and the gastrointestinal tract has to be adapted to retain the diet and the microbes to affect this fermentation. We'll spend the rest of this lecture, as we will the rest of this course, talking about herbivores. We'll talk about foregut fermenters, the ruminants and non-ruminants, and the hindgut fermenters, the cecal fermenters, and the colonic-cecal-colonic fermenters. To start with a familiar example, we'll cover the ruminants. We have here a sheep and a dik-dik, which is a small African antelope. These have an enlarged forestomach, as we know, which is a site of digester retention and microbial fermentation. Ruminants have a small to mid-sized cecum, which is a site of additional fermentation. For example, in the sheep, the cecum is reasonably well developed, and quite a bit of fermentation can happen in the cecum. As you've learned in earlier classes, the forestomach in the ruminant is composed of 4 stomach compartments, including the rumen, reticulum, omasum, and abomasum. The rumen participates in particle retention and microbial fermentation, and you can see it pointed out to you for the goat, sheep, cow, and the chevrotain. The reticulum is contiguous with the rumen, has the same function, and oftentimes these 2 compartments are simply referred to as the reticular rumen. The function of the omasum is not completely known, but it's thought that it participates in particle retention and water absorption. And the abomasum, the last stomach compartment, is the glandular stomach most similar to our own. Now, we've heard many times that ruminants have 4 stomach compartments. That's not strictly true. Not all of them do. The omasum is absent in the chevrotain, and we can see the chevrotain at the right. Chevrotains are referred to as primitive ruminants. They're thought to most closely resemble the ancestral ruminant. They're very small. They're the size of a large rabbit. Males have enlarged canines. This particular specimen is a female. You can't see them. Their diet is principally fruit, but if they see a mouse, they'll eat that as well. It's thought that they reflect a ruminant that existed before the omasum evolved. The ruminants are not the only herbivores that have an enlarged forestomach. There are non-ruminant foregut fermenters like the hippopotamus and the kangaroo that we see here. Some foregut fermenters have an enlarged forestomach with pouches like the rumen, like the hippopotamus. If you took off your glasses, the hippopotamus stomach may look something like a rumen. The forestomach doesn't always look like a rumen. It can be tubular like in the kangaroo. It almost looks like a colon. As in the ruminant, the last compartment is a glandular stomach. There is variable development to the hindgut. For example, the hippopotamus has virtually no development to the hindgut, whereas there is some development to the hindgut in the kangaroo. It does have an enlarged cecum and colon. Foregut fermentation is one strategy for herbivory, but there's another strategy, that of the hindgut fermenters. And one example of a hindgut fermenter is a cecal fermenter. We have a rabbit and a koala to illustrate cecal fermenters. These have simple stomachs, again very similar to our own. They have an enlarged cecum. You can see the cecum in the rabbit is quite enlarged. The koala has an enlarged cecum. The first part of its colon also appears to be enlarged, but we'll focus just on the enlarged cecum. This enlarged cecum is a site of microbial fermentation. It is unique in that it selectively retains fluid and fine particles, which are non-fibrous and highly digestible. It selectively excretes large particles, which are fibrous and poorly digestible. And those of you familiar with ruminants know that this is unique. In ruminants, it's the large particles that are selectively retained, and the fluid which is not retained selectively. So it's the opposite scenario in the cecal fermenters. Cecal fermentation permits small body size. Almost all herbivores under 50 kilograms are cecal fermenters. There are very few herbivores at small body size otherwise. The reason is that metabolic rate or energetic demands is very high, relatively speaking, for small body sizes. Metabolic rate scales with body weight to the 3/4 power, so metabolic rate is tremendously high per unit body mass for small animals, whereas it's relatively low for large animals. The relatively high metabolic rate for small animals presents a big problem. For herbivores, this is an especially big problem because their herbivorous diet, like grass, roughage, and leaves, is generally poor quality. The cecal fermenters are able to survive because their selective retention improves diet quality. It makes herbivory possible. They selectively retain the fluid and the fine particles, which are high quality, and excrete the large particles, which are low quality, improving their diet. Other kinds of hindgut fermenters are the colonic and cecal colonic types. We have here the pony and the African elephant as examples. These have a simple stomach, just like the cecal fermenters. They have an enlarged colon, which serves as a site of microbial fermentation, and a variably sized cecum, which provides additional fermentation. In the pony, for example, the cecum is relatively large. It's a cecal colonic fermenter. In the African elephant, there is no large cecum. It's strictly a colonic fermenter. The colonic and cecal colonic fermenters have no selective retention of fluid. The retention of fluid is the same or even shorter than that of particles, just like in the ruminants, but unlike the cecal fermenters, where the particles are retained much longer. These tend to be large, over 50 kilograms, for the reason that they don't have selective retention of fluid, and so small body sizes are not permitted. Foregut and hindgut fermenters have different advantages. Foregut fermentation is a strategy characterized by high efficiency. The diet can be digested more extensively because there's longer retention of digesta in the rumen versus the hindgut. Another reason for the high efficiency of foregut fermenters is that microbial products that are produced in the forestomach are subsequently digested and absorbed in the small intestine. These products include microbial protein and vitamins. In the hindgut fermenters, these products are lost unless coprophagy is practiced. In contrast, hindgut fermentation is a high throughput strategy. Hindgut fermenters are able to sustain high levels of intake. The forestomach in foregut fermenters restricts high intake because of long digesta retention in that site. The forestomach serves as a bottleneck. In ruminant nutrition, we talk about physical fill, limiting intake. This is what we're referring to. The hindgut fermenters have no such restriction, and so they can sustain much higher levels of intake of poor quality forage. As a result of these different strategies, foregut fermenters outcompete hindgut fermenters when food availability is limited and of high quality. High quality means low fiber, high protein. They make more efficient use of the limited feed, and there is no need for high intake when food is high quality. The hindgut fermenters outcompete foregut fermenters when food availability is high and of low quality. Low quality means that there's high fiber and low protein. There's no need to be efficient. High intake allows energy and nutrient requirements to be met even on the poorest diets. One illustration of this competition is shown on the African savanna. When the grass has grown tall, zebras are the first animals to come in and to eat the grass. It's of low quality, but it's of very high abundance, and so zebras do well. Only after the zebras clear the high grass do ruminants come in. They make better use of the regrowth, which is higher quality, but much more restricted in amount. In summary, anaerobes are sensitive to both oxygen and high redox. Low oxygen alone is not enough for anaerobes to grow. Carnivores, frugivores, and insectivores have the simplest gastrointestinal tracts, whereas the herbivores have the most complex ones, and that's simply reflecting the amount of fermentation that occurs in herbivores and the relatively little fermentation that occurs in carnivores, frugivores, and insectivores. Foregut and hindgut fermenters have different digestive strategies that bring unique advantages. The foregut fermenters are highly efficient, whereas the hindgut fermenters can sustain higher levels of intake.